Okay, so the first thing we're going to cover is how do you grow bacteria in the lab, and these are some actual physical pictures of how you might grow bacteria if you were doing research in the laboratory. So there's two primary ways that we grow them. One is in liquid culture. So this is an example of a test tube. This test tube contains liquid media, and within this liquid media are individual bacteria cells. So probably billions of bacteria cells are actually within this particular tube. The other way we grow bacteria is on solid culture. So this is an example of a petri dish, and this petri dish contains solid media. So this solid media is created from agar, which is a component of seaweed, but this creates like a hard gel type structure. So it's kind of like Jello, but a much harder version of Jello. And this Jello is infused with nutrients that the experimenter controls. And so when you use this inoculating loop, you spread bacteria, you spread liquid on top of this plate, and the individual bacteria can then grow on this plate. So what you're seeing is not the bacteria that has been transferred, but what you're seeing is the bacteria that have resulted from probably 24 hours of growth on this particular plate. And so, this little colony, this is called a colony, this little circle right here, that started from a single bacteria that grew into millions, if not billions, of bacteria over the course of time. So when you think about bacteria, one of the things you need to think about is what is their food source? What do they need to live? And each bacteria is different. Some bacteria are more generalists that are able to survive in different conditions. Some bacteria are specialists. They have to live in particular environments, maybe like a host that they're a pathogen for. So a lot of this is bacteria specific, and a lot of what we're going to talk about is related to E. coli bacteria. So E. coli is kind of the workhorse bacteria of most laboratories. E. coli is isolated from your gut originally. It prefers temperatures such as 37 degrees, but it's also able to live in a large variety of media conditions. And so the media contains an energy source, so both a way for it to produce energy, such as sugars. So sugars is a very common energy source that is used, and then it also creates all the essential nutrients and kind of the chemical molecules necessary for life. So this would include things like amino acids, nucleotides, fatty acids, carbohydrates, like all sorts of different molecules that are used to either create DNA, to create proteins, to create kind of whatever is needed. And so bacteria are probably pretty remarkable for they're like these molecular machines that are able to produce all of these different chemical components. And so there's two primary types of media that we're going to discuss. So one is rich media and one is minimal media, and that just tells you the extent of the molecules that are within this broth that is produced. So you could start from something as simple as minimal media, and minimal media contains salts. So these include things like magnesium, calcium. These are just elements that are necessary for any sort of life. So very simple types of salts, magnesium sulfate, potassium chloride, something like that. And you would have a range of these salts to kind of cover all of the different elements. And then you have a carbon source, so this might be glucose, and so the glucose contains carbon, nitrogen, oxygen, and hydrogen molecules, and then water. And so if you put E. coli bacteria within this minimal media broth, it's able to intake those salts, is able to intake that sugar into the cell, and then from there it uses its proteins in order to construct every single molecule that it needs. So it both takes that sugar and then it respires and metabolizes that into energy, into ATP, but it also uses that in order to make things like nucleotides, amino acids, so that it can produce more DNA, more proteins, more cell walls, etc. So there's hundreds of enzymes that are used for all of these molecular reactions that are encoded by the E. coli genome. You can also make it very easy on E. coli. So if you wanted to do that, you would use what's called rich media. And so this is media that contains kind of all of the chemical components of life. And so typically you would make this out of something like yeast. So you would grow up a lot of yeast, and then you would kill all the yeast, and then you would blend up all of this stuff into like a new kind of extract. And so, within, the yeast would have all sorts of proteins, all sorts of nucleotides, all sorts of sugars, all sorts of salts, etc. The bacteria would no longer have to make everything from scratch, but it could simply just import those molecules from the outside and then use them in order to produce DNA, proteins, etc. So rich media you should think as much easier conditions for bacteria. They grow faster in rich media, so they might double every 20 minutes versus every 60 minutes in minimal media, and the genetic requirements and the molecular requirements and the biochemical requirements are much simpler in rich media. And so if you have a mutant, you might have a mutant that has lost the ability to produce a nucleotide, and it would not be able to live in minimal media, but it would be able to grow in rich media. So in this case, you have liquid, and so you would put this test tube at a temperature like 37 degrees, because that's what E. coli prefers, since it was isolated from our gut. That's our body temperature. And you would put it in a shaker, so the shaker would make sure that the media remained oxygenated, that there was oxygen within there. And then this would eventually become cloudy because there are so many bacteria that grow within that media. And then you would also take a little bit of this liquid and you might put it on this plate and you might spread it around, and then you would create colonies. And so these are what I referred to before, these little circular parts, circular things. So there's two colonies right here. One of those colonies, each of those colonies grew up from a single bacteria. So all of these bacteria within the colony are pretty much genetically identical to each other, because they're all produced by this exponential process of one cell become 2 cells, become 4, become 8, become 16. And so if you wait long enough, you're actually talking about millions of cells that have been produced. And so often you go back and forth between these two types of growth. So you might take a suspension of bacteria cells, you take a small amount of liquid, you spread this suspension onto this petri dish that has agar gel, and then there's maybe 20 individual cells. So you pick a dilution of this culture, so there's very few cells within there, and then there might be approximately 20 cells, individual cells, that have landed on this plate. And then you wait from 1 to 2 days, and that one cell, each of those cells within that plate will have grown up into an entire colony. And then you also might use, this, go back and forth between the agar plate back to the liquid, and so you might have, a test tube filled with media, and then you would simply pick one of these colonies. So you'd have like a little plastic tip that you would do for something like that, and you would just lift up that colony, all the bacteria within that colony, and then you would just simply put it within the media within this test tube, and then again you would just incubate and then allow those bacteria to grow until they used up all the media within this liquid. Serial dilutions allow you to count the number of bacteria in a liquid suspension, and so here we just see if we add 100 microliters, 10 microliters, or 1 microliter, for example, of a very dilute culture, you can see you have a different number of colonies in each condition. So this allows us to count how many bacteria are within 1 microliter of this liquid culture. And so the idea is that every single bacteria that ends up on this plate is able to survive and is able to grow into this colony. So we simply go through and count 1, 2, 3, ... 4, 5, 6, 7, 8, 9, 10, 11, 12. So there would be a total of 12 colonies in 1 microliter of solution. And then, I'm not going to count here, but there should be approximately 10 times as much colonies on this, and then 10 times as much, on this, because you're adding, 10 times more and more. And of course, this is a stochastic process, and I kind of made up all these numbers, so I'm not even sure that these are actually 10-fold dilution. Sometimes you might do a 5-fold dilution or something like this, but this is the basic idea behind how you could count how many cells are actually within a liquid culture. And so the idea is that each colony on the plate represented a single bacteria within the liquid. All right, so how do you phenotype a bacteria? So each of these colonies come from individual bacteria, and each of these bacteria have a different genotype, could have a different genotype. So let's say we've mutated these bacteria so that there's new mutations within all of the different genes, and so some of the genes are affected in the different bacteria cells. And so, how do we phenotype? How do we associate that genotypic difference with some sort of phenotype? And so very often we use nutrient phenotypes. And so the idea is that mutant animals now require certain vitamins, sugar sources, amino acids, or something else to survive. So a prototroph is an organism, a bacteria that can grow on minimal media. So it can synthesize all essential organic compounds. So it's synthesizing nucleotides, amino acids, vitamins, etc. And it starts from a single sugar source that's provided in the minimal media. So it could be glucose, for example. An auxotroph, so a wild type E. coli is a prototroph. An auxotroph cannot grow on minimal media. So it either needs complete media or it needs minimal media supplemented with some sort of additional chemical molecule. And that's because it has lost the ability to grow, typically due to mutation, to synthesize essential compounds. So going from the sugar to something like methionine, it's lost one of the enzymes that's necessary for this. And if you take a biochemistry class, you'll cover kind of just how complicated these metabolic pathways are. But you might start out with glucose, and then that gets turned into another molecule, and then that gets turned into some additional molecules, and so on. And you end up having this huge web of different molecules that can be created from that original glucose molecule. But one of the end results might be methionine, which is an amino acid. And each of these steps requires an enzyme in order to convert this molecule to another molecule. And so this is where a genetic mutation can have an effect. And so if you've lost an enzyme that's required for creating methionine, then you can no longer survive. So if you put this bacteria on minimal media, it's not going to be able to grow. And that's simply because the animal isn't able to produce methionine, and so it's unable to produce any of the amino acids that contain methionine. And so that's essential, and so the bacteria simply dies. So some of the different categories, so for example, a vitamin, arginine, and methionine. So these are 3 examples of mutants that are unable, so this is the genotype right here, so bio minus, arg minus, and met minus. So these are 3 genes, and the phenotype is that they're unable to produce either a vitamin, which is biotin, I think that's vitamin B, one of the vitamin Bs, arginine, which is one of the amino acids, and methionine, which is another one of the amino acids. So this is kind of like an end result of a metabolic pathway. So what's cool about bacteria is that, if you take this minimal media that does not contain methionine and you add methionine to that minimal media, then you're able to rescue that phenotype. And so this is what this means, is that if you add methionine to minimal media, then the bacteria is now able to survive. So it's able to produce all of those other amino acids, all the other vitamins, and the single amino acid it's not able to produce, we've now added to the media, so it no longer needs to produce that on its own. And so the phenotype of a met minus strain is that it can grow on minimal media with methionine, but it can't grow on minimal media by itself. or it can't grow on minimal media supplemented with another amino acid like arginine. It has the requirement for this specific amino acid. And then arginine and biotin kind of follow that same logic, where you have to supplement the media with that molecule in order to allow that strain to survive. And so, how would you do this phenotype in practice? You would have a plate, an agar plate made with minimal media. You would have an agar plate made with minimal media plus methionine. And if you did a replica plating, you might find something like this. So we'll cover in the activity what exactly a replica plate is in more detail, but you should have read something in the chapter. But the idea is that you have a colony that grows in this condition, but not this condition. So we have a total of 5 different bacteria. 4 of them are not met minus. 4 of them can grow in minimal media or met minus. But this guy right here is met minus because it grows in these conditions, but it doesn't grow in these conditions. Some other phenotypes are sugar sources. There are a bunch of different sugars. So here we talked about glucose being a sugar that you start with. But let's say instead of making minimal media with glucose, you made minimal media with lactose. So lactose is just a different sugar. It's found in milk typically. And so the way these networks are constructed is that depending, no matter which sugar source you use, you're always able to get into the core network and then produce all of these different molecules. And so what happens if you knock out an enzyme that processes sugar very early on? So what you have is a phenotype where the animals cannot grow on a specific sugar. So this bacteria right here, which doesn't have this enzyme, would not be able to grow on minimal media with glucose, but it could grow on minimal media with lactose. And these are two sugars, lactose, and then galactose is another one. So this is a different type of phenotype where what's important is the source of sugar that you provide in the minimal media. And so you have these strains that can process some sugars, but not one of the sugars. And then finally, a very common phenotype that we'll talk about is the sensitivity to antibiotics. So antibiotics are chemicals that kill all bacteria pretty broadly. So they take advantage of conserved processes within all bacteria, such as cell wall biosynthesis, or some conserved parts of the ribosomes that are common among bacteria but are not present within eukaryotic cells. And so these chemicals target those really conserved proteins. And so streptomycin is just an example of one broad-spectrum antibiotic that's able to kill most bacteria. But you might evolve resistance to that antibiotic. So, one way is that you have a mutation in the protein that the streptomycin binds to. And so if that protein mutates, then you're no longer able to, the drug is no longer able to bind to it and inhibit its function. But you could also have kind of unrelated processes where you actually have a pump that's able to pump the streptomycin out of the cell. So like the streptomycin comes into the cell, but before it can bind to the ribosome in order to inhibit it, you immediately pump that back out into the extracellular space. So that could be another genetic change that gives you streptomycin resistance. But again, the way you simply test for this is you create media that either has streptomycin in it or streptomycin without of it. And by doing that, you can determine whether or not a strain is sensitive to that antibiotic. So again, just to kind of summarize minimal media, so the way you phenotype is you typically use minimal media and you supplement it or you change the sugar source. So you choose the sugar source, xylose, glucose, galactose, and that allows you to determine whether or not a bacteria can grow within these different sugar sources. So you assume that a wild-type E. coli bacteria can use pretty much any of the sugar sources. You can supplement with antibiotics and see if that kills the cells. And then if you have an auxotroph, you can supplement the minimal media with amino acids or particular vitamins, and then see if that rescues the effect of the auxotroph. And the end result is that the bacteria either grows or dies. You can also use rich media. So if you grow something on rich media, you can assume that the rich media rescues pretty much every single genotype. So this strain, so all of these strains right here can all grow on rich media. Because you can assume that there's methionine in the rich media, biotin. So all these things that they might need to uptake, they now uptake instead of producing it itself. And then you also assume that there's a variety of sugar sources. So like a gal minus mutant can live because there's other sugars besides galactose in that. And kind of the only exception is antibiotics. So if something is streptomycin sensitive and you add strep to the rich media, it'll still die. And finally, we're going to cover viruses as well, or phages. So viruses and phages are kind of used intermediately here, but a phage you can assume is a virus that targets a bacteria. So you can also grow viruses in the lab, and typically you're interested in phages that use bacteria, that infect bacteria. So these viruses, bind to bacteria. They inject their RNA or DNA and protein contents into the cell. They hijack the cell, and then they produce a large number of new phages, and eventually lyse the cell and release to kind of like repeat the whole infectious process. So how do you grow these in the lab? So you grow them using them as a bacteria food source. So you can either grow them in liquid, so you add phages to a liquid broth of bacteria, or you add phages to an agar that has bacteria kind of embedded within the agar. So this right here is just kind of like a side view of what that agar plate looks like, and you're assuming that kind of this entire agar is infused with bacteria cells, okay? And so now you do what's called a serial dilution, and so you dilute this liquid media that contains a large number of viruses, and you do that at different concentrations. So we're going to talk about two extremes first. So one extreme is that you add such a low concentration that there's no viruses that actually get transferred onto the plate. And so this is how the plate would look just normally. So you see like a uniform color, and all the bacteria within the agar are fine and dandy. The other extreme is that you add too much viruses, and so you kind of cover the entire plate with viruses, and all of the bacteria within the agar ends up getting lysed, and it changes the color and kind of the shape of the plate, so that you no longer see the color associated with the E. coli bacteria. But if you get the concentration just right, if you add a small number of phages, then what happens is something similar to the process that creates bacterial colonies. And so this single virus will kind of land in the middle, and then it will infect a bacteria. It will then lyse that bacteria and release more particles, and then those viral particles will start to spread out from that center, and it'll slowly be lysing the bacteria as it goes along. And so these are called plaques. So this again is the side view, and these plaques are basically where the viruses have landed, infected, and then start to spread their way out. So it's kind of like a process of spreading out. So this is now just like a physical picture, kind of right here, of one of these plates, and so each of these would be like a little plaque that's kind of associated with a viral particle. Plaques can also have phenotypes, and that is mainly due to some sort of feature of the plaque. So perhaps the color of the plaque, so this is turbid versus clear lysis plaque. and so you can kind of see that there's differences in how the plaque looks, and that's determined by the genotype of the phage. So the phage might spread in a little bit different ways. It might spread with, it might kill at a little bit different frequency, and so kind of how all that interplays changes how a plaque actually looks. But there's other things as well that you can phenotype, and so we'll discuss those more at the end of the chapter. But this is the end of part 1, and next we'll move into genetic exchange within bacteria.